Hi, so we will be talking about the host defense mechanisms or the ways in which the body protects itself. It is divided into two, non-specific and specific. The non-specific is further divided into two, which is the first line of defense and the second line of defense. On the other hand, specific is for the third line of defense. First line of defense, we have the skin, which is the physical barrier, also helps with the mechanical barrier. And if the skin is intact, the microorganisms cannot get into except when for certain helminths such as schistosomiasis and some hookworm diseases. Next is the mucous membrane. Some mucous membranes help with being a mechanical barrier for the cilia and the sticky mucus, which helps trap pathogens. So the mucus, it contains lysozyme. Lysozyme degrades the peptidoglycan in the bacterial cell wall. Okay, so it acts that way. And the lysozyme is also present in the sweat or perspiration. Second, we have lactoferrin. Lactoferrin, so the word ferrin, it's a clue that it binds with iron. And iron is acquired by all pathogens. So if the pathogens are deprived of iron, of this nutrient, they will not survive. They are deprived of this because they cannot compete with lactoferrin. Next is lactoperoxidase. What is lactoperoxidase? Lactoperoxidase is an enzyme that produces superoxide radicals. So ano po yung superoxide radicals? Our superoxide radicals is a highly reactive form of oxygen which is toxic to bacteria. Okay? Next, we have the cellular and chemical factors. So definitely, dryness, because bacteria loves moist, acidity to kill them, and also the temperature inhibit growth and colonization. Put a star, perspiration flushes the pathogens that has been trapped away. So again, it contains the lysozyme which degrades peptidoglycan in bacterial cell wall. Next is the microbial antagonism. Microbial antagonism, it's the competition for colonization sites or nutrients. But if it is not effective, superinfection can occur wherein there is a decrease of number of indigenous microbiota at a particular anatomical site, leading to, let's put a big arrow here, overgrowth of pathogens, such as opportunistic pathogens at the site. Just some additional for the first line of defense, thousands of bacteria are removed from the oral cavity every time we swallow. Okay, so all the dirt there, okay? And at the GI tract, digestive enzymes, stomach acid, and the alkalinity of the intestines play their vital roles in protecting the body. Next, for the second line of defense, we have transferrin, fever, interferons, complement system, inflammation, and phagocytosis. Alright. So first off, we have the transferrin. Okay. So for the transferrin, same with the lactoferrin earlier, the word ferrin, no, it's a glycoprotein synthesized in the liver. And it deprives pathogens of iron. Okay. The normal function of transferrin is to store and deliver iron to host cells. So, just think of lactoferrin and view it as similar. So, if the iron binds to the transferrin rather than to the pathogens, the pathogen is deprived of the nutrient, so the pathogen will die. Next, we have fever. So, with fever, can you remember the term pyrogens or pyrogenic substances? So these are the substances that produces fever and may originate either outside of the body or, let's say, inside of the body. Okay, so outside of the body, what we have here are any pathogens or anything that releases endotoxins are pyrogenic substances. And then for inside of the body, one example that we have is an interleukin-1 or IL-1. Okay, so from WBC, this is an endogenous pyrogen. Next, we have interferons. So when we say interferons, it interfere with viral replication. So interferons are small antiviral proteins produced by virus-infected cells. And there are three known types of interferons. We have the alpha, beta, and gamma interferons. For the alpha interferons, we have the B lymphocytes, monocytes, macrophages. For beta interferon, we have fibroblasts and other virus-infected cells. And the gamma, we have activated T lymphocytes and NK cells. Okay, please remember at the start that interferons are not virus-specific but host-specific. So for example, there is a rabbit interferon. The rabbit interferon will work with rabbits but it will not work with humans. Interferons are also the ones that produce or cause non-specific flu-like symptoms such as malaise, myalgia, chills, and fever. So this is still under the second line of defense. So we have the complement system, which is a group of 30 different proteins. The actions are complementary to the actions of the immune system. Kaya nga tinawag na complement system. Okay, so let's draw a ladder here. So complement components or the proteins of the complement system, what they do is that they interact with each other in a stepwise manner. So this is what we call the complement cascade. So as they target the AG, AB, or antigen antibody complexes, which is the lock and key in an analogy, this is what happens. So first is opsonization. Okay, so in opsonization, they tag enemies for attack. Okay, so they find the target, and there is also a deposition of what we call opsonins. So these opsonins, these are complement proteins that coat pathogens to make them easier for phagocytes to recognize and eat, such as macrophages. Okay, so basically opsonins are the one that tag them or mark them. Okay, opsonins, these are antibodies or certain complement fragments. Next is the MAC or the membrane attack complex. Okay, for the membrane attack complex, they punch holes in for the invaders, and the one responsible for this is the complement protein C5a to C9 to form the MAC. And again, what they do is that they drill holes. Okay, so they drill holes in the membrane of bacteria or infected cells to let the pathogen burst and die. So next thing that occurs due to the complement system is the stimulation of inflammation. So just recall your cardinal signs of inflammation. So rubrica, dolor, tumor, and functionalisa. So what are the pathways involved in the complement system? First, we have the classic pathway. So it's about the immune complex and AG-AB complex, and it starts after the adaptive immune system makes antibodies. So the classic pathway, it needs antibodies. Next, we have the mannose-binding lectin pathway. So in the mannose-binding lectin pathway, no antibodies are needed. And it is part of the innate immunity, and it is triggered by lectin binding with the mannose groups in bacteria. So that is why it is called mannose-binding lectin pathway. Next, we have the alternative pathway. And this is triggered by contact with various viruses, bacteria, fungi, and tumor cells. So this happens automatically on foreign surfaces. So no antibodies, no lectin needed, just triggered by the pathogens. And so before I forget, opsonization, what causes it, or what are the proteins responsible for it? It is the complement protein C3b. Okay, so this connects all the complement pathways. And yeah. Now, how about the chemotaxis?
So, chemotaxis is the directed movement of a cell or organism in response to a chemical stimulus. So, you think of a taxi along a C5 highway because the protein for chemotaxis is C5a. Now, how about cytokines? So, your cytokines, they are chemical mediators that are released from many different types of cells in the human body. And in short, our cytokines, these are what we call our chemical messengers. So imagine, so there are two cells. So one cell With the presence of the cytokines, it will be able to talk to another cell because cytokines enable cells to communicate with each other. Fifth is about the inflammation. So with inflammation, we have already discussed this earlier, we called the cardinal signs. So with the inflammatory response, there are three major events. First is vasodilation. Second is the escape of plasma and plasma proteins due to the increased permeability of the capillaries. And third is the escape of leukocytes and accumulation at the site of injury. So what is the purpose of the inflammatory response? So this is of course to localize infection, to prevent pathogen spread, to destroy and detoxify pathogens, and to aid in repair and healing. Last, in the second line of defense, we have phagocytosis. So with phagocytosis, it is about surrounding and engulfing, that's our keyword here, or ingesting foreign materials. So now we talk about the third line of defense. The third line of defense is about the production of antibodies. And when we say antibodies, this is produced in the body in response to presence of foreign substances. Okay, and these foreign substances, these are what we call your antigen. So it's already in the word, antibody generating. Okay? So an antigen must possess several properties for it to be immunogenic. Okay, when we say immunogenic, that's the term for substances that are capable to induce an immune response. So this means that not all antigens can produce an immune response. So we have to look at the immunogen and look at it like, yes, I can induce an immune response. So that's what we call immunogenic. So the B cells and the T cells, they look for the immunogenic part of an antigen. So that's what we call the epitope or epitope. So this is the specific part of an antigen that is recognized by the immune system. So it acts as a precise docking site where an antibody B cell receptor or T cell receptor binds to detect a foreign invader. Okay, so for example, we have proteins. Okay, so proteins are most immunogenic because they are larger and they are more complex structures. For example, egg allergy. So the egg white contains ovalbumin, which is a protein. Okay, that's why a lot of people has egg allergy, right? So now we try to compare glycoprotein versus pure protein. Glycoprotein is more immunogenic than a pure protein because a glycoprotein is a protein that has sugar molecules attached to it, while a pure protein consists strictly of amino acids without any added sugar chain. So this means that the glycoprotein is more complex, so it's more immunogenic. The molecular size of antigen affects its immunogenicity. So the rule is if the molecular weight is less than 10,000 Daltons, it is weakly immunogenic. But if the molecular weight is greater than 10,000 Daltons, it is very potent. But also consider the stability of the molecule. For example, a molecule with an increased molecular weight but breaks into a smaller molecular weight once they enter the body, they lose their immunogenicity. Also depends on how the antigen is administered. For example, when an antigen is administered intramuscularly, there's no reaction. When an antigen is introduced subcutaneously, there's a good response.